Good morning, everybody in California, and perhaps those who have joined from the eastern part of the globe. It's a good night time, I hope. So this is the first screen, and what you are seeing are two artificial brains talking to each other. And in this presentation, we will try to understand how could we invent a more sophisticated version of EEG. EEG stands for electroencephalogram. So it is there are some probes that you put on your on your scalp, and you take the potential and you try to understand. We believe that the signals that we measure from the scalp is actually coming from the brain. And mm, uh, mm, 1875, this instrument was first made by Richard E. Caton, and it has been going on. So far, people have tried to increase the frequency from 1 to 40 hertz and 40 hertz to 300 hertz. Uh, uh, if we try to measure higher frequencies, then there is a problem. The problem is that there are a lot of electrode polarization, and we cannot do the measurement. But the problem with EEG is that um, it measures it measures even could be the muscle signals or other kind of signals which has millisecond vibrational time domain. So this is a slide. We did this work 2012, 2013. We were measuring microtubule. Suppose microtubule is lying, and you put electrode horizontally and vertically. That means orthogonal to each other. And you, just like you imagine you have a basic transistor-like systems, but you have, instead of one gate, you have many gates. And you have a source and drain. If you do that for neuron, for microtubule, for tubulin, yeah, as you know, the neuron is a cell in the brain, and it is considered as fundamental to our thought process. And then if you go inside the neuron, you find microtubule that is a nanowire, 25 nanometer diameter, or hundreds of micrometers long. And if you go inside microtubule, you find tubulin protein. So at that point of time, we try to understand the resonance frequency. So normally, when physicists measure resonance frequencies of a particular biomaterial, they put two probes or four probes and pump electromagnetic signal and see the transmission coefficients at which frequencies maximum power is transmitted from one side to another side. But in this case, the experiment was unique in the sense is that we discovered theoretically that that both sides. I mean, suppose I have an object, say my mobile phone, and if we send a signal this way or this way, there could be a 2D surface profile, and that is what exactly you are seeing here in the screen. So what happens when you change these frequencies? You see the higher dimension of the resonance band. So this is a beautiful, apparently it is a very simple experiment, but if you think about it, it's a beautiful way of understanding much more in much more depth the frequencies. And you see triplet of triplet. That means you see three bands of frequencies, and the horizontal and vertical. And in those regions, those set of combination of frequencies actually activate the entire material significantly. So you see three bands, and each band contains three different frequency regions where activity is much more. So triplet of triplet we observed. And then I forgot this work. In 2020, one of now he is currently my friend, Josh Neo, actually. You see in the bottom, 2020, he was doing the measurement, and he found triplet of triplet in his brain. And when he takes it out, the probe takes it out, and then he finds that there is no signal. Apparently, this is a very crude, very crude method. But when you see this kind of thing, the signals are coming out. As you know that EEG could be measured not just in the brain, but it could be measured all over the body. So the signal comes out from all over the body, and maps were there, papers were published in the early days of 1900, when people are trying to understand how credible it is. We have also done similar kind of studies. What tells us this particular experiment, and I was amazed to see this experimental results, and then I informed Stuart that this has come, so why not we do it. So we studied hundreds of human subjects afterwards, and we made many different kind of very sophisticated instrumentations and technology we developed, because it is not so easy to measure the signal from the brain. So today it's a very short presentation, but I will try to make you understand what are the technological challenges over there and how did you overcome. Because if you want to understand details, that neuroscience needs revolution, what Thomas was telling in that conference lectures, you can see details. But more and more we are understanding this particular high frequency signal that is coming from the brain. I would like to get into that. So I'm not getting into details of how it has been confusing. If you take EEG, because if you blink your eyes, if you move your hand, then EEG signal changes, because membrane resonance frequencies and then EEG frequencies that you see there, they are almost similar. So 40, 50, 60, the heart, this particular region is very sensitive, both the signals. So whether you are seeing the muscle signal or other kind of signals, is very difficult to isolate them. And also over the time, because of the huge commercializations, these are the 6 points: cleaning and artificial removal. So I have copied this kind of problems associated with EEG from different research activities. You can search in the net. There are fantastic papers who have delved into the realistic features of EEG. What I would like to tell you that there are enormous amount of research that has been done, which says that neuron firing is ultimately eventually giving rise to EEG signal. If you say that, then you are forced to accept, when I would say that from the brain I'm getting microsecond or nanosecond time domain signal, that must be coming from the neurofilaments or protein complexes or other kind of thing, but not the neuron firing, because they happen only in the millisecond time domain. So let me get to this. So one particular message that I would like to deliver today is that we say DDG. Why DDG? In duodecanogram. That means when we are measuring the human subjects, large number of human subjects, we found that from millisecond to we go to the picosecond. I mean 5 to 6 seconds, where we use the thermal camera to find out the signals. When you go to that region, we find 12 different time domain where activities are more. So our proposal is that brain activity is not limited to the millisecond time domain, and we are saying that it is in the microsecond or nanosecond. We are saying that you have to look, if you want to understand a cognitive processes or perceptions or other kind of things, then you have to look into different time domain signals, and you can find a higher level pattern that is emerging, and that could be the proper way of looking to it. So where we are technologically different and what we are doing. So two different modes we are taking the signal for DDG, high frequency time. Look, you can't with the EEG machine, you can't look into high frequency signal. It's absolutely you can't do it. So one way is to send, say, picosecond signals, pulse streams we can send, and we can have a logic analyzer connected to it and next to it. And as you can see in the diagram, whenever there is a nanosecond burst, say 20 nanosecond burst, then the logic analyzer get active. It counts, and then potential drops. It also drops. So this is one mechanism by which we calculate that data. Another is we have an antenna and directly measure the radiations that is coming from different part of the brain. So there are two modes which work simultaneously in the DDG machine. Now you can ask that, Anirban, why do you take two different types of measurement? In EEG, we are simply looking at the potential or current flow that is there on the surface. Because personally, I don't believe that EEG is giving us the real brain signal. If there is a high frequency signal, a part of it will remain confined on my brain surface, and a part of it should come out. So I need to check, because if it is coming deep inside from my brain and it is limited on my brain surface, it is very unlikely as a physicist to think that it would remain on the surface and not come out. So to make it, to understand that it's really a not an artifact on the brain surface. I mean, I want to go back to 1875, that point of time when we are making it. Surface current is flowing on my brain, and that is coming deep inside from my brain, penetrating the skulls and coming to the top. Fine, fantastic. But that would remain within the on the scalp and it will not come radiating out. That's very difficult. That's very difficult to accept for me. So not just we look at the potential bursts that are happening, and in case of EEG, what you do is with every microsecond, say, or hundreds of microseconds, you take the signal, change in the potential, and you create the EEG, as you can see in the schematic. But but that doesn't tell you the proper time for which the signal is generated in the brain. So that problem is actually addressed in DDG when we are making it, and then we measure the radiation also. The third thing that we did is we created absolutely replica. Today we don't have the time, but we have other presentations of the CIHS. Where you can see that how did we created the brain. So now I will jump on, because of this short time, to the results. How does the DDG results look like? So this is our brain model that let me bypass all these things. Artificial brain, because as a physicist, we always, when we do the measurement of two device, suppose I have a device, I want to measure a semiconducting property. I have identical another device, and then we compare both the devices with respect to each other. So that's the standard physics or engineers practice, because you can't understand the property of a single device if you measure the single device alone. You need to have an identical device, identical connections, identical circuitry, identical instruments, except instead of this material A, you could have empty, or you could have a very dumb material that is over there, whose properties are very well known, simple, and then you can do it. Why not we do it for DDG or EEG experiments? So that's why in the artificial brain, we were using one human being, one artificial brain. Artificial brain is absolutely direct replica from single molecular scale to the large neural network, almost like the human brain. And we put it and then normalize it. So that means it could be helpful for us in future to understand the human consciousness and human responses, because we can normalize. So now you see that EEG and DDG, that is there, you have to do some more technological changes. So apart from the electrode design, two mode simultaneous interaction, what is coming out of the surface and the surface current simultaneously together, to understand if one increases, other decreases, then we can at least say that, okay, might be it is passing through the skull and then coming out. Apart from that, if you look at it, in case of EEG, at the two different backbones are the point one or two, depending on which company's EEG machine you are using, you normalize the signal, okay? But in DDG, you have to do, apart from those two, you need to have three more. One is the clock that we have here. You have seen the little babies when they are born, when the babies are born, then you have seen that this part, there is nothing on the top frontal part of the head, and that is a beep, 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 beep. You can see the heartbeat. So that is one clock here, and then the blood vessels coming out, and they also create huge pulses in the brain, because if you look at the brain with your heartbeat, with respect to heartbeat, whole brain vibrate, boop, 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 like this. And it's a massive vibration of the brain. Since it is normalized, we don't feel it, but you can see there are some images over there. But if you want to do the DDG measurement, high frequency measurement, that particular normalization, when blood's entering into your brain, and then it is colliding on the higher surface of your skull. So that particular difference in the extreme high pressure, and blood is electromagnetic. And there are lots of metals, and then if it is flowing, then it generates electromagnetic fields also. So that effect needs to be neutralized to understand what is the signal for the actual brain. So these are the three fundamental changes we made. And when we are looking at it, then we create, you see number C, just below in my screen, you can see that there are many electrodes, and and we see a tensor. So it's a tensor that actually changes the activity of over time. So you get spatiotemporal dynamics, and that dynamics we study. So unlike EEG, where you are looking into you are looking into just the signals that is coming in a particular frequency range. Here we look into multiple different time domains simultaneously, because that is what we found different. I repeat it. In EEG, when you are taking the whole brain, different spaces, how the currents are coming, and then say 6 to 8 hertz, what would be the possible activity regions you find. In DDG, of course, we take that, but we take many of them, and then we try to figure out how, over time, a particular dynamics, a particular geometric shape is emerging, to find out the invariants. So many of you know invariant, E is equal to mc square. So so m is a variable, E is a variable, and c square is the invariant. So if you need to know invariant and the variables, how the variables are connected, to understand the fundamental of any phenomenon. So if I want to understand thought process of my brain, then also I need to know the invariant. So that is what we did with the DDG system. So as you can see that we made several changes uh, in the technological development. So we put 1 person, 2 persons, 3 persons, 4 persons to 8 persons, 9 persons, 10 persons, 12 persons, up to 12 persons we went. We put kilohertz, megahertz, gigahertz, terahertz, many different kind of machines. It was a massive network in the room. And we try to understand that do we need one human or two humans or more humans to understand, because ultimately we need a marker for emotion, marker for cognition, marker for different kind of cognitive features. So 91 different cognitive features we did. So the results on the surface, it almost look like EEG. So Martin teams created this kind of softwares. I mean, this is very easy to create. It is not, I mean, only the physical principles. So the engineering is novel, but this kind of software is also the other kind of EEG softwares you can use for the analysis. And this is kind of how the lab look like. So we separately checked where the gigahertz signal is more, or nanosecond time domain activity more, where it is. And then we found that every single human being has a very peculiar time domain. And then we try to classify human subjects into different categories. And we could found that 8 to 9 different categories of humans are there, who have a very unique profile mapping on the brain, but we need to study more to generalize it. And this is kind of a snapshot. So in case of EEG, on the top, you can see that there are some EEG patterns. And then you can go to higher milliseconds, microseconds, nanoseconds. So 84 snapshots we take at a time, and then we try to find out which region activity is more. And then we try to understand the activity. And then we try to find out if there is a geometric shape of activity regions connected, remain independent or unchanged. Okay, so And so we found that when the number of human subject increases, we can, when we reach 8 human subjects, we are able to find out 3D geometric invariants for particular cognitive properties. And so invariant, I wanted to tell that invariant is in a radius. If an object is moving around the circle, then radius is invariant. And physicists always look for invariants. um, I, I do want to avoid this. Uh, the real data, how does it look like? It's too complicated. I will bypass. You can see the CIHS presentations, but only thing I would like to say is that if you play the music and you allow the brain to see the signals, when you put the room with smell, the activities is huge, and a human's majority of the cognitive processes, cognitive things actually happen in 200 to 900 700 milliseconds, so 10 megahertz to 120 megahertz. Around this particular frequency domain, we see the activity, though we go for gigahertz or nanoseconds or picoseconds. It is not there much. I'm not claiming that we should look into this frequencies, but still I'm saying that we should look at all the time domains. And after 150 years of EEG, that should be our goal. So we have tried to find out invariant shapes, and the study is going on. It's not complete, but uh, we are still trying to understand and trying to make a better and better device. There is no limit, as I can say that I would like everybody to join the DDG movement and try to see how can we contribute, because I think that after 150 years of EEG journey, time has come for us to look into this entire time domain and try to invent invariants. We, as a singular group, these are all the members who have worked for the human subject studies. A large number of human subjects were studied, but why do we need a large number of people or researchers to work together to get it done? Is because there were a huge number of instruments that were there in the circuitry and cables connected to the brain, because we are looking at, say, multiple brains at which frequencies they synchronize. If everybody synchronizes, we consider that as a credible data. One human being, no, because we wanted to understand that irrespective of race, caste, creed, and nationalities or whatever, for a human, for a particular cognitive experience or perception, there should be only one geometric shape responsible. With that, I would like to thank you for giving me, Thomas and Jeffrey, to give me the opportunity to present my work, and 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 I think that I've finished in time, and then we can go for the next presentation.
Yeah, thanks so much, Anirban. Maybe while Stuart is setting up his presentation and stuff, looks like Gino might have a question. So
yeah, I guess this might be a little moot because of the different bands and everything. But just looking at the different brains, I guess with Peter Fennick, and actually we've been doing some work too in hyperscanning. I know we just have it in the we don't have it at the very high frequencies, but is there anything that we can draw from that, really looking at two brains and intersubjective engagement? Thank you.
Yeah, so basically we have seen that we have reported it. Recently the paper came out. When you have more than one subject, and whether they will synchronize or not, we don't know. We cannot give a guarantee. So this is absolutely raw experiment. Where in many of our experiments, we put 8 persons and they absolutely don't synchronize at all. So the first question needs to be asked, will that 2 human beings synchronize? Synchronization at millisecond time domain when EEG is very easy, but synchronization at a higher time domain is very, very difficult, especially at a different time domain. Another thing, is that if they synchronize, now I would like to answer your question. If they synchronize two persons, and how and why they would synchronize, that knowledge we don't have. We are at a very primitive level. I mean, most of concentrated on the engineering part we have concentrated in our research. So if two humans being synchronized, then what happens is, suppose you play music, you will find part of the music is played in one person's brain and part of the music is played in the other person's brain, and we have seen it repeatedly. It is so beautiful. And if you have 8 persons, the technology, everything I have shared, is everything free, and anybody can contact me to understand, to get this, the circuitry and everything. It's already published, and then some papers and more are coming out. I can share the wiring. You would be able to see that, suppose you are playing a music, for an example, with Shiva Stotram we played. You play some beautiful, say, Tibetan music, which is extremely harmonizing. And after 15-20 minutes, the two persons get synchronized. You will find some part of the music is played in one's brain, some part other, as if tick, 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 at least different time domain, different brain is getting selected, and they are coming up. For the first time in your life, you would realize that your consciousness is not alone, and you will see it live. And I have seen it. I mean, it's so blissful and so spiritual to see. And when you play, and what you do, if you do that, next time when you do EEG or whatever, fill the room with smell. Fill the room with smell and do that. You see magical changes. And another thing, we have it, and you can go to CIH's presentation, which is much longer and much more detailed. You will see, if you give some food, take some sweets, both of you, and look at the brain, look at the signal. You will see desynchronization, and hold hands, left hand, right hand, and you check the signal. You will see dramatic changes. So touch, then smell, then food. And we kept the room dark, nearly dim. Okay, so we also gave visuals. So every single sensory system has a very unique thing, but human consciousness have something to do with smell. You will see it very sharp, very active, and very unique synchronized response that you will get from